Hi everyone, so today we'll be covering the June 2023 paper 1. As you can see here, this is from the Edexcel Biology A-Level B, and you can see the code for the syllabus here. So this is paper 1. This paper takes about 1 hour and 45 minutes, and the total marks for this paper is 90, which is about 30% of your final grade. Now, this paper covers these following topics that you can see here. Let me just paste that so you can see. All right, so it covers topics 1 through 7, as you can see here. And let's start off with the first question. Okay. All right, question 1. Plants take up inorganic ions from the soil. These ions include calcium, phosphate, and potassium. A1. Name the molecule that contains calcium ions in the cell walls of the plant. So this would be calcium pectate. Let me just write pectate. So this material is found in the middle lamellae. Let me just show you an image of that. So here we have two adjacent plant cells. We have your cell wall here, and then we have the middle lamellae here. And this pore here is the plasmodesmata, so that is a pore that links the two cells together. And your middle lamellae is where you would find your calcium pectate. Okay, the next part. How many of the following statements are correct? Let's go through them one by one. So phosphate ions are used to make DNA and RNA. That is correct. Let me just show you the general structure of a nucleotide. So if you recall, your nuclide consists of a phosphate group, pentose sugar, and this is the nitrogenous base. Let's go over the second sentence. Phosphate ions are used in the structure of the cell membrane. That is correct. So if you recall a phospholipid, you have your glycerol, your two fatty acids, and then you'd have your phosphate group, which is your phosphate ion, and that's a part of the phospholipid that is hydrophilic. And the last sentence says the element potassium is part of the peptide bond. That is incorrect. So this is correct, correct, this is incorrect, because a peptide bond would be this here. All right, so the answer is 2 only. Part B, the uptake of potassium ions by plant seedlings in anaerobic and aerobic respirations or aerobic conditions was investigated. The seedlings were grown in a mineral ion solution containing a low concentration of potassium ions for 30 hours. The conditions were anaerobic for the first 15 and aerobic for the second 15 hours. Okay. So it was anaerobic. So let me just write this down. Anaerobic first, and then it was aerobic for the second 15 hours. So one of the things that we can mention is, why is the concentration of potassium ions decreasing and why is it increasing? So we could say that perhaps it's decreasing because maybe potassium ions are being used in the cell by certain metabolic processes, or you can say the reason why it decreases is maybe it left the cell. And the reason why the potassium ion concentration is increasing is because it entered the cell. So let's write that down first. So there is a decrease in the potassium ion due to the potassium ions leaving the cell, the increase in potassium ions is due to it entering the cells. Then we need to talk about what kind of processes occur for these things to happen. So in the beginning, in the first 15 hours, it was anaerobic, and the concentration of the potassium ions decrease. So it must be some sort of mode of transport that is passive, that does not require energy. And that would be diffusion, but it's not just any kind of diffusion, it would be facilitated diffusion. So facilitated diffusion is basically when charged ions or polar molecules like glucose would go from a region of high concentration to low concentration using a protein channel. Because potassium ions are positively charged, it can't just go through the membrane. It has to rely on a protein channel. So let's write that down. We left the cell and then let's talk about the second 15 hours. So in the second 15 hours, you see that it is increasing. And in the second 15 hours, aerobic respiration occurred, which means that this mode of transport must have relied on energy, on ATP, via aerobic respiration. So that means that in order for the potassium ion concentrations to increase, it must rely on active transport. So let's write that down. So the ions left the cell via facilitated diffusion, but it entered the cell via active transport. As oxygen was available for aerobic respiration to occur and produce ATP. Okay, let's go through the marks. So the first mark is the decrease of concentration of potassium ions is due to it leaving the cell, whereas the increase is due to it being taken up by the cell or entering the cell. So that's one mark. The second mark is the ions left, or the ion decreases by facilitated diffusion. That's the second mark. And the ions are taken up by active transport. So that would be the third mark. And then the fourth mark would be oxygen is available for aerobic respiration. And the last mark, the fifth mark, would be that aerobic respiration generates or produces more ATP. All right, let's go on to question 2. Living organisms are divided into 3 domains: Archaea, bacteria, and eukaryota. The table shows some characteristics of organisms in the domain Archaea. Okay, so here's a little bit of history. So Archaea used to be part of bacteria, and there used to be a 2-domain system in which you just had bacteria and eukaryotes. But then afterwards they found out that Archaea had features that are quite similar to eukaryotes and also very similar to bacteria as well. So now we have a 3-domain system in which Archaea is now its own domain. So let's go over the traits one by one. So here we have membrane enclosed nucleus, and that's absent in Archaea. So that is very much similar to our bacteria, because bacteria don't have a nucleus. Now, peptidoglycan and cell wall is absent, so bacteria has cell wall made out of peptidoglycan, whereas eukaryotes don't. So this is very similar to eukaryotes. And let's say the third one. So third one, 70S ribosomes. This is very similar to bacteria, whereas eukaryotes have a larger ribosomes, and we refer to them as 80S ribosomes. So we can say it is not classified as bacteria because they do not have peptidoglycan in their cell wall. And then we can also say it is not classified as eukaryota because it does not have a nucleus and has 70S ribosomes. Alright, so let's go over the marking scheme. So 1 mark for not bacteria because they do not have a peptidoglycan in their cell wall. And the other mark is not eukaryota because of not having a nucleus, or you can also refer to the 70S ribosomes. Alright, let's go on to part B. Alright, so it says some Archaea have the enzyme adenylate kinase. This enzyme is similar to the AK enzymes found in humans, and there are 2 types of this enzyme in humans, AK1 and AK3. The table gives some information about these 2 enzymes. You can see that there. So the difference between AK1 and AK3 is the location and also the substrate, but the roles are still the same. So for part 1, it says the tertiary structures of AK1 and AK3 are very similar, but not identical. Analyze information to deduce why there are 2 AK enzymes. So the reason why they are very similar is because they have the same role, and that is to transfer phosphate. So let's write that down. So the structure is similar because they both have the same function, which is to transfer phosphate. And so the reason why they would not be identical is because their substrate are different, which means that their active site would have a different shape. So let's write that down. They are not identical because they have different substrates, which mean that their active site is, or has a different shape, has a different shape. So this will actually give you 3 marks, but you can also elaborate on the location or where you can find these enzymes in the cell. So because they're located in different places of the cell, then they would have different structure. And in the marking scheme, it also says the pH might be different, or the pH is different in the cytoplasm and the mitochondria, and that's why it has a different structure. So let's go over the marks. One mark would be they're similar because they have the same function, which is to transfer phosphate. That's one mark. The second mark is they have different substrates, and the third mark is the active site has a different shape. Okay, let's move on to part 2. Some types of Archaea have only one type of AK enzyme. This enzyme can catalyze both the reactions shown in the table. The tertiary structure of this enzyme is different from AK1 and AK3. Explain why Archaea have a different type of AK enzyme. So if you look back at the table, the reason why humans need two is because of the location. One's in the cytoplasm, the other one's in the mitochondria. And Archaea have a very similar structure to prokaryotes in the way that they don't have mitochondria. So that means that the AK enzyme would just be found in the cytoplasm. So we can say Archaea do not have mitochondria, so they do not need two enzymes. Another thing is Archaea needs to be able to have these two different substrate fit into its active site. So that's why the structure would be a little bit different. So sorry, I spelled that wrong. Archaea would need an active site that binds to, or that can bind to ATP and GTP. So some other marks that are on the marking scheme is you can say that Archaea's chemical environment, like the pH or the temperature, is different to that of humans. That's another thing that you can say. One more thing you can also say is the DNA sequence is different in Archaea versus in humans. And because you have a different DNA sequence, then that means that your amino acid sequence would be different as well. All right, so let's just mark these 2 points here. So Archaea do not have a mitochondria, so there's no need for two enzymes. That's one mark. And another mark is that the active site needs to be able to bind to both ATP and GTP. All right, let's move on to question 3. Okay, question 3. Xylem and phloem tissues transport molecules and ions through the plants. Part A, give the meaning of the term tissue. So tissue is just a group of cells that work together to serve a same function or that perform a similar function. So we can say a group of cells that perform similar function. All right, part B, the table gives some information about the structures of xylem and phloem tissues. For each statement, put one cross in the appropriate box in each row to show whether these statements are true for both types of tissue, xylem tissue only, phloem tissue only, or neither. Okay, let's go through them one by one. So let's look at the first row. It says contain sieve plates, and that is only present in the phloem, as you can see here. So this would be the sieve plates that are found in the sieve element of the phloem. The next part is have cellulose in the cell wall. That is true for both of them because they're both plant cells. And then mitochondria, that would only be true for the phloem. The xylem is pretty much hollowed out cells, hollowed dead cells. And then your mitochondria would be present both in the sieve plate element, a sieve tube element, and the companion cell. So that would be phloem only. All right, part C. Aphids are insects that can be used to study the transport of sugars in the phloem. The diagram shows how this is done. So we have step 1, the leaves of the plants are covered with a glass chamber and contain radioactive CO2. So the CO2 would be here, and then it would undergo photosynthesis to produce radioactive sugars that would go down the phloem, right? And then we have aphids A, B, C, and D, and the aphids are analyzed to determine the time at which the radioactivity first appears in their bodies. So part 1, which disaccharide is transported in the phloem? That would be the sucrose. All right, the next part, part 2. In an experiment, the time between radioactivity appearing in aphid A and aphid D was 210 minutes, sorry. So aphid A is here and aphid D is here. Let me just draw that out here. So we've got A, D, and that took about 210 minutes. The distance between these two aphids were 50 centimeters. Okay, so what we would do is we would figure out the distance and divide that by the time taken in hours. First, we got to convert the minutes into hours by dividing that by 60. So 210 divided by 60 would give us 3.5 hours. And then we would do 50 divided by 3.5. Oops. And that would give us 50 divided by 3.5. That would give us 14.29 centimeters per hour. Part 3, describe how sugars are transported through the phloem. All right, so this is how sugars are transported through the phloem. So I've included a diagram here. So sugars are transported by mass flow. We have the sucrose molecules that are in the source. In this case, we have a leaf cell. So a source is just part of the plant that is producing sugars or releasing sugars. And what happens is that the sucrose is transported to the phloem via active transport. And so what happens is that water will want to move into the phloem via osmosis, and that would increase the hydrostatic pressure over here. At the same time, at the bottom, because you have a higher water potential in the phloem compared to the xylem, water will leave the phloem to the xylem, and that causes a decrease in hydrostatic pressure. And because of this difference in hydrostatic pressure, sugars will want to move from a region of higher to lower hydrostatic pressure, and then the sucrose will move out of the phloem and into the sink. So let's write that down. So I should say sucrose are transported via mass flow. First, the sucrose is transported from the source to the phloem via active transport. This causes water to enter the phloem via osmosis, and that increases the hydrostatic pressure. At the same time, near the sink, water would leave the phloem and into the xylem, which causes a decrease in hydrostatic pressure. Sucrose would move, so sucrose would move to a region of lower hydrostatic pressure, which is the sink, then it would move of the phloem, which is, I should say near the sink, then it would move out of the phloem and into the sink. All right, let's go over the marks. The first mark is mass flow. The second mark is sucrose is moved to the phloem via active transport. The third mark is sucrose moves out to the sink. And then the fourth mark is describing how water enters the phloem. Water enters the phloem via osmosis. This happens at the source, which I spoke about in the previous sentence. And then it leaves the phloem near the sink. So that would be the fourth mark. And the fifth mark would be sucrose would move to a region of lower hydrostatic pressure. All right, question 3 is done. Let's move on to question 4. Okay, influenza is an infectious disease caused by a virus. The influenza virus is an RNA virus that has an envelope. Which pair of viruses also have RNA and an envelope? So this part of the syllabus, you need to memorize this. I've included a table here which summarizes these two features across these 4 viruses. So these 4 viruses are part of your syllabus. Let's go through the options one by one. So Ebola does have RNA, does have an envelope. HIV does have RNA, does have an envelope. So the answer is A. With tobacco mosaic virus, it has RNA but no envelope. With lambda-phage, it has DNA and no envelope. Okay, let's move on to part B. Which of the following is the predominant method of transmission of the influenza virus? So this would be respiratory droplets. Right, so things like sneezing, for example, or coughing is another one as well. All right, let's move on to part C. Symptoms of influenza appear sometime after a person is infected with the virus. Explain why there is a time delay between infection and the symptoms appearing. So the reason why there is a time delay between infection and the symptoms appearing is because it takes time for the viruses to replicate and to infect a large population of your cells in your body, which is when the symptoms start appearing. So that's one mark, and the other 2 marks would basically be talking about any part of the lytic cycle. So I've included that here. Okay. So you can mention things like, first the virus would need to attach to the cell, or it needs to insert the genetic material into the cell, or it needs to be able to synthesize the proteins that it requires for it to form the virus the virus particles, or you can say it takes a while for the viruses to assemble the different proteins together to form the final virus particles. All right, let's just remove that. So the reason why there is a time delay is because it takes time for the virus to infect and damage a large population of cells. And then again, we can mention the different steps in order for them to make the virus particles. So again, referring to the lytic cycle. So we can say, example, it would take time for the host cell to synthesize the viral proteins and to assemble to form new viruses. Okay, let's go over the marks. So time is taken because it takes a while for it to damage a large population of cells, and another one, take time for the host cell to synthesize the viral proteins and to assemble them to form new viruses. Those are the 3 marks. All right, part D. In some countries, people take antibiotics without a prescription. In a survey, 77% of people who had taken antibiotics had taken them without a prescription. Some of the people who took antibiotics without a prescription had influenza. Part 1. Calculate the ratio of the number of people who took antibiotics without a prescription to the number of people who took antibiotics with a prescription. So I'm just going to write without versus with. And if I had 100 people, then 77 people would have taken antibiotics without a prescription. And then to calculate the number of people that took it with a prescription, you would do 100 minus 77. That would be 23. Now we want to simplify this ratio. So we divide both sides by the smallest number. So therefore, this is going to be 1 here. And this is going to be 3 point, and it says 2 decimal places, so 3.35. 5 to 1. So in the marking scheme, they calculated it different ways. Well, this answer is still accepted, but you can, in this case, because we divided by the smallest number, in the marking scheme, they've divided it by the largest number. They've divided it by 77 instead. But the answer is still correct. Okay. The last part, explain why antibiotics should not be taken by people with viral infections. So antibiotics are for bacteria. And when antibiotics will not work on viruses because viruses aren't even cells to begin with. So that's 1 mark there. And the other mark would be, if you were to use antibiotics unnecessarily, it would serve as a selection pressure for antibiotic resistance bacteria. right? And then you would end up having a larger population of antibiotic resistance bacteria. Okay, so let's write that down. So antibiotics do not work on viruses because viruses are not cells, right? And the other mark is the unnecessary usage of antibiotics serve as a selection pressure causing the increase in antibiotic resistant bacteria. So let's go over the marks. Antibiotics do not work on viruses because they are not cells or they are not living. And the second mark would be a selection pressure causing an increase in antibiotic resistance bacteria. All right, so we've done 4 questions so far, and we'll cover the last half of this paper in the next video. Bye!